everyone, and welcome to today's webinar, Current Innovations in Cancer Organoid Modeling for Drug Discovery. I'm Christy Jewell of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more about our sponsor, please visit thermofisher.com. Now, we encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for at the end of the presentation. Also, if you experience any technical questions, you can utilize this Q&A box and we will assist you directly. I'd now like to welcome our speakers. Presenting first, we have Dr. Colin Paul, Cell Biology R&D Staff Scientist, Thermo Fisher Scientific, and Dr. Thierry Jarde, Laboratory Head, Department of Anatomy and Developmental Biology, Monash University, Australia. For complete biographies on our speakers, please visit the Biography tab at the top of your screen. And with that, I'd like to invite Dr. Colin Paul to start with his presentation.
I'm excited to present how Gibco OncoPro tumoroid culture medium can enable tumoroid-based compound screens. The OncoPro medium kit, shown here, is designed for the culture of patient-derived tumoroids from epithelial cancer indications, including colorectal, lung, head and neck, pancreas, endometrial, and breast cancers, and makes physiologically relevant patient-derived tumoroid culture more accessible and standardized. Our interest in patient-derived cancer models has been driven by the recognition that, while cancer is responsible for around 10 million deaths worldwide each year, development of an anti-cancer therapy typically requires around 10 years and $1 billion US. Some of these costs occur because the majority of compounds that look promising in preclinical research fail during clinical trials. Today, we'll focus on the use of physiologically relevant patient-derived tumoroid models in the development of anti-cancer compounds, which could include small molecule inhibitors, cytotoxic drugs, targeted protein degraders, biologics, antibody-drug conjugates, and hormone therapies. As these compounds are evaluated functionally for their effects on cancer cells, the development of accurate and representative in vitro cancer cell models is crucial for understanding tumor biology and improving therapeutic strategies. A number of the approaches discussed here can also be used in the evaluation of potential cell therapies with these cancer models. Traditional immortalized cell lines have limitations in mimicking the complexity of patient tumors, including properly representing gene expression patterns and clinically observed mutations. Primary patient tissue in animal models provide greater biological complexity but can be difficult and expensive to source and maintain. In contrast, patient-derived tumoroids, or cancer organoids, are obtained directly from patient samples and grown in 3D culture systems using specialized serum-free media such as OncoPro tumoroid culture medium. Compared to immortalized cell lines, tumoroids more accurately reflect the biology and heterogeneity of patient cancers, have scalable growth, and importantly, retain the genetic and phenotypic characteristics of the original tumor to enable use throughout the drug discovery pipeline. Today, I'll discuss 5 steps to enable tumoroid-based compound screens, including sourcing tumoroid models, culturing and characterizing tumoroid cells, optimizing conditions for compound screens, and running assays using different analysis modalities. Let's step through each of these in more detail. Step 1 is to source tumoroid cells for use in compound screening assays. I'll focus on examples of colorectal cancer, or CRC tumoroid lines that were derived from human cancer cells using OncoPro tumoroid culture medium. Our typical workflow involves dissociating fresh resection samples, aggregating cells in suspension for 2 to 7 days, embedding tumoroids in basement membrane extract or BME domes for initial growth, and then expanding tumoroids in suspension culture for bank generation. Additional reagents for handling tissue and deriving tumoroid lines are listed here, and Thermo Fisher has an application note available that describes this process in more detail. While new tumoroid lines can be derived from tissue samples, we also offer a number of Gibco OncoPro tumoroid lines across cancer indications that have documented 3D morphology, genetic and phenotypic stability, and consistent growth profiles, and can be used to quickly implement physiologically relevant tumoroids in assays of interest. During tumoroid line derivation, a common challenge using culture media systems designed for adult stem cell organoid culture is the expansion of non-cancer cells. These healthy cells can sometimes overtake cancer cells in culture. OncoPro tumoroid culture medium was designed for the expansion of epithelial cancer cells and designed to not promote healthy cell growth. And we've seen from flow cytometry and RNA sequencing data that cancer cells are selectively supported over time. Stromal and immune cells, for example, that are present in the initial tumor samples eventually die off, as you can see by decreases in CD45 and CD31 protein levels by flow cytometry between the initial tumors and established tumoroids. Importantly, tumoroids derived in OncoPro tumoroid culture medium retain donor-specific mutational profiles and gene expression patterns. Targeted sequencing using the OncoMine Comprehensive Assay V3 shows that both the presence and frequency of single nucleotide variants and key oncogenic mutations are retained when comparing initial tumor samples to derived tumoroids across a range of genetic backgrounds and different mutations. The plot on the left shows matched single nucleotide base substitutions and oncogenic mutations between colorectal tumor samples and derived tumoroids, where mutations are consistently conserved in culture. Additionally, RNA sequencing reveals a high concordance of gene expression levels between initial samples and tumoroids across indications. The heat map on the right shows a comparison of tumor samples to tumoroids for select colorectal, lung, and endometrial cancer donors. Overall, we see a typical correlation in gene expression patterns between starting material and tumoroid cells of around 85%, and most of the difference is due to the loss of immune and stromal cells over time. After tumoroid cells are growing stably in culture, they can be expanded and banked. OncoPro tumoroid medium was developed for compatibility with a suspension culture approach, where tumoroids are grown in 3D in the presence of a small amount of basement membrane extract protein, but handle much like a suspension cell line. They do this while retaining the morphology and gene expression patterns that are observed in embedded culture within hydrogel domes. And a lot of our culture involves some of the reagents listed on the bottom right of this slide. This enables generation of large banks of tumoroid lines, which we typically feed every 2 to 3 days and passage every 4 to 14 days. And we call these lines established once they have these stable growth rates, demonstrate at least 5 cumulative population doublings from the original cell number, and are bankable and freeze-thaw competent. Importantly, the morphology of these tumoroid lines are donor-specific, as you can see on the images on the left, and maintained over passages in vitro. These also grow at stable rates both before and after cryopreservation, which is indicated in the plot in the middle of this slide by arrows for freeze-thaw cycles. And in many cases, this growth is stable over 250 days. These lines also exhibit distinct subpopulations within the tumoroids as measured by single-cell RNA sequencing to show evidence of tumor heterogeneity. After scale-up, we freeze tumoroids in recovery cell culture freezing medium to establish working banks that can be pulled from for downstream assays. Characterization is very important for these patient-derived tumoroid models. The key draw of these models is that they look like patient tumors. We frequently perform next-generation sequencing to compare early tumoroid banks to tumoroids that have been cultured in vitro for longer periods of time. In many cases, this involves use of the Ion Torrent OncoMine Comprehensive Assay for mutational profiling and the Ion AmpliSeq Transcriptome Human Gene Expression Panel to analyze gene expression patterns, as well as routine monitoring by microscopy and at times flow cytometry. This type of analysis has demonstrated high concordance between early banks of tumoroid lines and cells in culture, in many cases for up to 250 additional days. Correlation analysis of the frequency of single nucleotide variants, or SNVs, identified using the OncoMine panel show high correlation. These are all at 0.98 and above, between tumoroid banks at passage 13 and tumoroids after 53 to 258 additional days in culture. Additionally, this is a nice fingerprinting approach, as you can see that there's very little cross-correlation of SNV patterns between donors. Consensus molecular subtyping from gene expression data additionally demonstrates that colorectal cancer subtypes are maintained in a donor-specific manner long-term. You can see 3 donors on this slide. That occurs at different passage numbers as shown here. And a diversity of these different molecular subtypes are supported by the media system. Principal component analysis and differential gene expression analysis tell a similar story. Global gene expression patterns are conserved during long-term culture, with tumoroids at multiple passages in the PCA plot on the left clustering by donor and not converging on any common phenotype. Additionally, differential gene expression analysis between cultures at passage 13 and at passage 50 show significant changes in less than 3% of over 17,000 genes, and these changes are associated with minimal significant changes in KEGG pathways. Overall, we've seen that tumoroid lines established using OncoPro tumoroid culture medium are quite stable in culture. Optimization of tumoroid cultures for downstream assays involve both scale up to generate cell numbers required for different assays and scale down, typically to 96 or 384 well plates to achieve the required throughput. We typically use non-tissue culture treated flasks for scale up. These flasks are well suited to suspension culture with OncoPro medium and are available in a number of sizes. In particular, the suspension culture approach in standard flasks enables researchers to generate the cell numbers they need when they need them. Tumoroid morphology and growth rates, as population doublings per week here, are consistent from T25s up to T175s and triple flasks, and millions to tens of millions of cells per flask can be harvested to dissociate and seed into well plates for screening and analysis. This lets researchers maintain the biological relevance of tumoroid cultures while benefiting from the ease of use of the OncoPro medium system and the suspension culture approach. We routinely employ suspension culture to generate cells for downstream assays instead of having to individually plate tens to hundreds of basement membrane extract domes during scale up. After scaling up, we'll typically dissociate tumoroids and seed cells into 96 or 384 well plates for high throughput testing. Measurements of cell viability after plating show low variance across plates for multiple tumoroid lines and indications, and this variance can be further minimized by incorporating automated fluid handling during cell seeding, as you can see in the plot on the left. Overall, our guidance is to anticipate needing around 1.5 million cells per plate, with 12 to 15,000 cells per well in a 96 well plate and 3 to 5,000 cells per well in a 384 well plate. Formats such as microcavity plates, which include micropattern divots at the base of each well, can be used to further control tumoroid size. As far as best practices, we typically will strain cells through a 100 micron mesh cell strainer prior to plating and after dissociation to remove any non-dissociated cell clusters and promote even well-to-well seeding. One other watch out is to minimize cell settling during handling by using agitation or trituration, as these tumoroid cells tend to settle out of suspension more quickly than immortalized cell lines. Our typical timing involves seeding dissociated cells, culturing for 3 days to form tumoroids, and then incubating tumoroids with compounds from day 3 to day 7. Images of tumoroids at day 3, the time of compound addition, are shown here. At this time point, we observe low well-to-well variability across a given plate after following the cell seeding best practices discussed on the previous slide. Step 5, now that you have cells in a plate, is to run assays to detect and measure cell viability in response to compounds. I'll show examples of plate reader-based screens, which provide rapid readouts on a per-well basis, as well as high-content image-based screens to gain more information on cultures and improve amenability to space and time-resolved analysis. Thermo Fisher Scientific offers a number of instruments and reagents to facilitate cell analysis, including the various ScanLux multi-mode plate reader and the Cell Insight CX7 laser high-content analysis platform. One reagent that has worked particularly well for us in 3D cultures is called out on this slide, the Cell Event Caspase 3.7 detection reagents, in which a fluorescent signal is activated by caspase 3.7 activity during apoptosis and is compatible with both live and fixed cells. As an example, one plate layout we've used for a compound screen in a 96 well format is shown here, though other configurations are certainly possible and may be better depending on the experimental situation. Our typical layouts involve serial dilutions of compounds with analysis across multiple tumoroid lines, with 2 to 3 wells per condition, as well as positive and negative controls. I'll show a pretty simple example using staurosporine in both plate reader-based and image-based assays in the next slides. For plate reader-based assays, we prefer to multiplex orthogonal measurements of cell viability to assess dose response to compounds by a few different methods. Assays can be run in about a week, as shown in the flowchart at the top of this slide. One analysis protocol involves incubation with Presto Blue HS or high sensitivity viability reagent, which is most accurate for 3D models if it's incubated overnight. You can then read that plate and then sample the supernatant to quantify lactate dehydrogenase release from dying cells via the cyclone LDH cytotoxicity assay. And finally, lyse the cells remaining in the plate to quantify ATP levels at the different treatment conditions. Some example dose response curves to staurosporine using patient-derived colorectal tumoroid models, as well as HCT 116 immortalized colorectal cancer cells, are shown on this slide. One thing that's been interesting is that we see a highly heterogeneous response with these physiologically relevant tumoroid models. The HUCO 031721 model, shown in orange, for instance, shows marked resistance to staurosporine treatment, while the other tumoroid models generally are more sensitive than HCT 116 cells. In general, dose response trends are comparable across assay readouts, though IC50 values can vary depending on the mechanism of action of the detection reagent. I won't spend much time here, but one nice thing about the OncoPro medium system and its built-in suspension culture approach is that it's quite amenable to automated fluid handling. We can run these assays using an automated liquid handler and achieve comparable response as when we run the assay manually, but we gain the benefits of having reduced hands-on time with automation approaches. We typically perform similar multiplexing for image-based fluorescence detection of cell viability. Staining with cell-permeable nuclear dyes identifies all cells, while combining both cell event caspase 37 green with Imagit TMRM reagent distinguishes apoptotic and alive metabolically active cells, respectively. Other dyes can also be added depending on the instrument setup. We found that these live cell compatible dyes give the most consistent results when they're co-incubated with cells and tumoroids for multiple days prior to refreshing the stains and imaging around day 7. This type of approach, when combined with high content analysis on platforms, such as the Cell Insight CX7, shows really clear distinction between live and dead cells when the dose of the compound is changed. So you can see these multicellular tumoroids changing from red to green on the images here as staurosporine concentration increases and apoptosis occurs. Additionally, imaging reveals heterogeneity in drug response, with both live and dead cells observed at intermediate drug concentrations. These dyes can also be read on plate readers equipped for fluorescence detection, but you can see from these viability curves that markedly better signal-to-noise ratios and this really nice crossover between live and dead cell populations are observed when image-based approaches are used. So for some of these in-depth analysis routines and experiments where you really want to know what's going on with the state of different tumoroids within a given well, we really like these image-based approaches. In summary, we're excited about the continued growth of tumoroids as relevant target populations in functional assays during drug development. And we feel that OncoPro tumoroid culture medium, some of the lines that we've established, and some of the protocols that I've pointed out here will make these types of experiments more accessible to the research community. So let's conclude. Tumoroids can serve as a more functionally relevant target in functional compound screening assays by representing a diversity of donor genetic backgrounds, having compatibility with automation, and then using multiplexed assay readouts to gain confidence in the results. A lot of the information I presented here is presented in more detail in an application note that's also available. Should mention that custom services, as well as the OncoPro tumoroid lines that we've established, can accelerate the use of tumoroids in different applications, and we'd be happy to discuss potential projects there. Going forward, we anticipate that tumoroid models will be used more and more in high-throughput compound screens, as well as functional genomic screening. And these types of models have already shown great promise in functional screening assays, even to guide cancer treatment selection. We're really interested to see how these may move more towards the clinic in the future. Overall, Thermo Fisher is excited to support your tumoroid work throughout the workflow, really from sourcing, culturing, characterizing, engineering, and assaying these tumoroid models. I'll wrap there, and thanks very much for your attention.
Thank you, Dr. Paul. And now I'd like to welcome back Dr. Thierry Jarde to begin his presentation.
Thank you very much. Good afternoon, everybody. Please bear with me while I'm sharing my screen. Well, good afternoon, everybody. My name is Thierry. I'm laboratory head in the Department of Anatomy and Developmental Biology at Monash University in Australia. I'm also the director of the Monash Biomedicine Discovery Institute organoid program. And it's a real pleasure to be here with you today on Zoom. And the purpose of my presentation is really to give you an overview of the breast cancer organoid technology, what we've done to really validate and characterize in detail these breast cancer organoids and make sure they replicate the features of the primary tumors that they are derived from, how we can use these breast cancer organoids for drug testing, and finally showing some of the early data that we're generating using the OncoPro culture medium. So I'm going to start with explaining why we study breast cancer. And indeed, breast cancer Sorry, trying to move to the next slide. Can you see my next slide? Yes. So indeed, breast cancer is an extremely common disease in Australia and in the world. We know that 1 out of 8 women will be diagnosed with breast cancer in their lifetime, which represents nearly 20,000 new cases of breast cancer diagnosed per year in Australia. And in the world, we're expecting more than 2.3 million people will be diagnosed with breast cancer in 2024. Unfortunately, we know that it's a very lethal disease, and in Australia only we have 8 breast cancer patients that died on a daily basis, which represent more than 3,200 patients a year in Australia only, and more than 600,000 deaths in the world. So what is quite frustrating with breast cancer in general is the fact that when the disease is diagnosed very early, when breast cancer hasn't had a chance to invade other organs, or when it's restricted to the breast, the chances of being cured are very, very elevated. So for a stage 1 tumor restricted to the breast, the survival rate for breast cancer patients within 5 years is 100%. In contrast, for advanced breast cancer, where the breast cancer cells have migrated to the lymph nodes and potentially to other organs such as the brain, the bones, the liver, well, this is what we call a stage 4 disease, and this is associated with very low patient survival. And overall, we know that for the 5-year survival rate for patients in stage 4 breast cancer is less than 25%. So how do we treat currently advanced breast cancer? So we know that the main approach will be to do a breast surgery, followed by radiation therapy. And finally, in order to target, potentially some of the cells that have had a chance to migrate to other organs, we apply chemotherapy. And one of the most common treatments is called, the ACT regimen, that would combine doxorubicin, cyclophosphamide, and paclitaxel. This type of chemo is currently given to, advanced breast cancer patients. But I guess what is the main limitation of this approach is this is what we call the one-size-fits-all approach, where we give the same drug to a cohort of breast cancer patients. And what happens in this context is that we know that if we've got an example, of 10 advanced breast cancer patients, we know that all tumors within breast cancer patients are different. They have different molecular subtypes, they have different set of mutations. They also have different cell populations with very specific microenvironments, such as specific immune cells or fibroblasts, which makes these tumors either resistant to the treatment or sensitive to the treatment. And for these advanced breast cancer patients undergoing standard treatment with chemotherapy, we know that the treatment response rate is 50%. So for this 50% of the patients, there's a clinical benefit. So there's either a complete response, therefore, the mets are gone, or a partial response, which means the tumor decreases in size. However, For the other 50% of the patients, there's no clinical benefit. So the disease keeps progressing. However, the patients are still under, and still going through quite severe side effects. So the question here is, what can we do to predict whether patients' tumor cells are going to respond to chemotherapy or not chemotherapy? And we are proposing the development of a precision oncology approach where we would identify breast cancer patients and breast cancer cells that are likely to respond to chemotherapy. And another group of patients in this context, the red patients in red, would benefit from another type of treatment. And how do we put that in place? Well, because this is an organoid symposium presentation, of course, we believe that implementing and developing breast cancer organoids for breast cancer patients and performing drug testing can really help us identifying drugs that are efficient. So if we focus on breast cancer organoids, and we contributed to at least the identification of the growth factors that were required to sustain breast cells in culture. This model, really the breast cancer organoid model, was described in 2018 by Sachs et al. In the Netherlands in Hans Klüter's lab. They've shown that if you take a tumor from a breast cancer patient, that you dissociate at the single cell level, and that you plate this single breast cancer cells into a 3D matrix, they very rapidly self-assemble with the right growth factors into these structures that we call the patient-derived breast cancer organoids. And what we were very keen to do here at Monash University was to develop a biobank of breast cancer organoids. And therefore, we developed a network of clinicians, surgeons, pathologists, and scientists to use this model to potentially predict, use this tool as a predictive tool for personalized therapy. So we've established this pipeline of tissue collection in collaboration with surgeons and clinicians at Cabernet Health, in collaboration with Dr. Gary Richardson here in the middle. Then at the Monash Biomedicine Discovery Institute, we've got multiple scientists that are working on these key questions. But we have also an organoid program team that is really focused on generating these organoids. And I would like to mention these two persons, Dr. Jasvina Kaur, here at the back, and Dr. Delis Lund, that are very essential in developing these breast cancer organoid models. And finally, we are fortunate enough to have a fantastic pathologist, Dr. Tim Nuttall, that really help us identifying the cancer cells. So what is really the workflow that we have at Monash? So once a breast cancer patient is diagnosed with a tumor, there will be, first of all, surgical removal from our surgeon, Dr. Peter Gregory. The pathologist, Tim Nuttall, takes slices of the tumor and provide these slices of tumor to us at the Monash Biomedicine Discovery Institute. We collect the tissues, dissect these tissues into very small pieces using a tissue chopper, digest using a cocktail of enzymes or collagenase and dispase to go into the single cell level. And we eventually seed these cells in a 3D matrix. And for our work, we used the Matrigel from Corning. And I know there are multiple other matrix that are used in the field. For example, the BME type 2 can also sustain organoid formation in this 3D environment. So over the years, we've generated more than 47 breast cancer organoid lines. And these pictures here are really representative pictures of 15 different organoid lines from patient number 1, so human breast cancer 1, up to human breast cancer number 22. And you can see here that these breast cancer organoids have different morphologies, probably associated with their proliferative rates and their molecular subtypes. This movie here is showing you an example of an organoid line that we've grown successfully over the 6 day period of 6 days. Sorry, and this is really showing that you can expand these organoids in 3D, dissociate them further, and then re-expand. So you've got access pretty much to a cell line, and this is now considered as a gold standard in the field. So what is very fascinating about this model is that we can generate organoid line from patients directly, which is unique really in the field. But we can also generate these organoids from multiple molecular subtypes, because we know that breast cancer is not an homogeneous disease. And at least there are 4 different subtypes that have been identified in the field: the luminal A subtype, the luminal B, the HER2 subtype, and the triple negative subtype. And for all these different subtypes, we've been able to generate organoid lines. What is important to remember here is the culture conditions that we are using are more likely to sustain the luminal tumors and the growth of these luminal tumors than the growth of the HER2 and the triple negative subtypes. So it's a work in progress, and we are really hoping that in the future we'll be able to generate organoids with a similar success rate across all these subtypes. So what have we done at Monash, really in Monash Uni, to characterize these breast cancer organoids? So we've decided to use a multi-omics approach by combining multiple different methods to characterize these organoids. But due to time constraints today, I will only focus on 2 points that I believe are essential. I will discuss the data that we've generated by whole exome sequencing, where we can compare the mutation profile of the primary tumor with the breast cancer organoids and the matched organoids. I will also discuss briefly our single-cell RNA sequencing data to discuss cellular heterogeneity across multiple lines. And finally, I will show you and provide some examples that we can use this bioresource to perform drug testing. So if we start with the whole exome sequencing data, this heat map, so we've done this work across 16 primary tumors and 16 of their matched organoids. We've identified the mutation profile of these samples, and this figure, this heat map, is really showing you whether the samples are related or not related. And indeed, what you can see here on the left-hand side, you've got the scale. Right. So when there's no relation between samples, it should be dark blue, and when there's a very strong relationship between samples, it should be orange. And what you can see here on the right-hand side is the sample identity. So for example, here we've got HBC41, so patient 41. This is the PT, the primary tumor, and the one below is organoid. And we've done that for all the different patients on this y-axis, and it's the same for the x-axis. What you can clearly see is that the organoid and the primary tumor for patient 41 really nicely correlates with, patient 41. So there's a very strong correlation between the organoids and the primary tumor across the entire population of samples that we assess across these 16 samples. So there's a very strong relatedness between the organoids and the primary tumor. But when you start looking into the details here and looking at very specific mutations, you can find a slightly different results. This figure is showing you the number of driving mutations on the y-axis. So for example, PI3K or P53 mutations that are known to be driving breast cancer progression. And we are showing you in orange the mutations that are found solely in the organoids, the mutations that are found in primary tumor, or that are shared between the organoid and the primary. And you've got very good examples here, for example, the line number 2, 5, 14, or 28, where all the mutations that are driving mutations are shared between the organoids and the primary tumor, suggesting that they very nicely mimic the mutation profile of the primary tumor. However, you can also find a couple of samples in these sets where, for example, HBC17 and HBC41, where the majority of the mutations are found in the primary tumor, and there's a very limited number of mutations that are shared between the organoids and the primary tumor. And indeed, if you look at this table where I've listed a set of genes at the top that are known to be mutated in breast cancer, you've got on the left-hand side the ID of the sample, so HBC16 primary tumor or HBC16 organoids. And if we look at a very good example where there's very nice retention of the mutation in the organoids for HBC14 here, In this example, you can see that the mutations are very nicely found in the organoids and mimic the mutations of the primary tumor. So HBC14 in this context is a very good model. In contrast, when you look at HBC41, the primary tumor, you can find a list of mutations that are listed here, including mutations in PTEN or mutation in TP53. However, these mutations do not appear in the organoids. So how can we explain that? So I'm just going to show you this data that we've generated recently, and this was done by our bioinformatician in the program, Dr. Stuart Archer. He generated these river plots that are showing you the presence of subclones within breast cancer organoids and within the primary tumor. So if you look on this figure here for HBC5, on the right-hand side, you've got the primary tumor. And in this primary tumor, we've identified 3 subclones, the blue clone, the red clone, and the green clone. And these clones are driven by a specific set of mutations. So the blue clone is essentially driven by AKT1 or ANKA2 mutations, while the green clone, for example, is driven by an HDAC4 mutation. What is quite nice here for HBC5 is that in the organoid, so this is the schematic in the middle, you can see the presence of these 3 clones. So based on mutation profile for HBC5, you've got a very nice retention of clones, of clonal diversity. When you look for HBC36, on the right-hand side, you can see that we've got 3 distinct clones in the primary tumor that are driven by CACNA1D or PMS1 or TRNP1. When you look at the organoids, you can see that the blue clone is maintained. However, there's the appearance of this orange clone, which is driven by MIB, which was potentially very limited in cell number in the primary tumor, which really suggests in this context that with the growth factors that we are using, it's very likely that we might be able to enrich for specific subclones, or in contrast, lose some subclones that are not going to grow in these conditions. So overall, I think the take-home message here is really to suggest that if you want to use the breast cancer organoid technology and organoids in general, it's very important to perform whole exome sequencing characterization to make sure that your model really mimic the mutation of the primary tumor. I would like to really discuss now the single-cell, our single-cell RNA-seq approach. And we've done single-cell RNA sequencing across 6 breast cancer organoid lines. We've got these 6 lines that are listed here. HBC5, 20, 35, 36, and 41 originate from luminal tumors, and while HBC14 is a triple-negative tumor. And what is very obvious here is that if you look at HBC14, you've got a presence of 1 cell cluster. And this blue cell cluster is not present in any of the other luminal lines, really suggesting that we are able to maintain unique molecular characteristics of triple-negative tumors. When you look at the luminal tumors, the other 5, you can see that for HBC5 and HBC35, they share a similar number of cellular clusters, so very likely have a similar cellular composition. But for HBC 20, 36, and 41, you've got the presence of these 3 main clusters, the light blue, the red, and the green, suggesting that they are more likely to share similarities across these 3 different lines. So overall, this figure is really showing you that we can maintain heterogeneity across multiple lines. And then when we focus specifically on a 1 line, for example, HBC14, you can see also that we can maintain cellular heterogeneity within a line. Because in this context, you can see that for HBC14, we've got the presence of 6 different clusters. And it's really mimicking what you would observe in primary tumors, where it's expected to have a very nice, a very high number of cell populations. So I've shown you that breast cancer organoids are heterogeneous and also maintain mutations of the primary tumor. So is it possible to use this model that seems to mimic very nicely primary tumors to perform drug testing? So I'm going to give you a couple of examples from the lab, and I'll finish with one paper that was published in 2021. So what we've done in the organoid program in my lab was really trying to develop this concept of personalized medicine. And we started by using organoids that were established from patient 5, 30, and 35. These 3 patients were diagnosed with advanced breast cancer and therefore were treated with doxorubicin. And we asked the question whether, first of all, we could perform drug testing on these organoids and whether we could identify differences between these organoid lines. Therefore, we plated these organoids into 96 well plates and exposed them to various concentrations of doxorubicin ranging from 0.001 to 10 micromolar. And we assess cellular viability at the end of the experiment at day 6 using the pressed blue assay. What you can clearly see when you look at these curves is that for patient 35 here in green, you've got a line that seems to exhibit a profile of resistance where you need very high concentration or higher concentration of drug to lead to a decrease in the number of cells. While in contrast for HBC5, so in blue, this is the opposite. These lines seem to be more sensitive than HBC35 to doxorubicin. And indeed, when you look at the IC50 for these 3 patients, so this is the concentration of drug that allows 50% of cell death, you can see that for HBC5, you can achieve this IC50 using a concentration of 0.2 micromolar, but for HBC35, it's 0.17 micromolar. So you need 10 times more drug for HBC35 to induce a similar phenotype. So with this system, we can identify, organoid lines that seem to be resistant to drugs versus organoid lines that seem to be sensitive to the drug. And what we are aiming to do in the future is really compare this in vitro data to in vivo data to the patient data and see whether there's a correlation. More recently, we've decided to use an approach where we can identify potential drugs based on their mutation profile. So we were really interested in FGF receptor targeting and really looking at a specific target called FGF receptor 4. And this study was published in Breast Cancer Research in 2021. So we screened for all the organoid lines that we generated for FGF receptor 4 and identified 2 lines, the HBC22 line and HBC30 line. HBC22 express very high level of FGF receptor 4, while the HBC30 do not express this receptor. We then decided to demonstrate that we can target FGF4 receptor using an inhibitor. And as you can see here, these are the 2 lines treated with the FGF4 receptor inhibitor or with the control. And you can see that only the HBC22 line was sensitive to this treatment, while for the HBC30 line, there was no impact. And therefore, with this approach, and these are the quantification that we did using the cell viability assay, the pressed blue, you can see that there's a significant difference between the FGFR4 expressing organoids versus the non-expressing organoids. So based on the mutation profile, you can potentially identify targets that you can use and druggable targets to potentially predict, or improve patients' outcomes. And I will finish with this concept of personalized medicine by mentioning this paper that was published by Shen et al. In Advanced Science, really pushing the boundaries of personalized medicine. What they did was to, they had this patient 148, which was a triple negative breast cancer patient with a primary tumor that was found in the breast and some micromets in the lymph nodes. They started by doing chemotherapy, so a neoadjuvant chemotherapy before surgery. And this chemotherapy, it's a combination here of carboplatin, docetaxel, and cyclophosphamide. However, with this treatment, the disease was still progressive, so PD means progressive disease. So the team decided to do a surgical resection followed by radiation therapy. Unfortunately, 7 months later, the same patient relapsed and they identified multiple lung metastases in January 2017. So they provided another drug, another chemotherapy called capecitabine. However, with this drug, the disease was still progressive and the tumor was still growing. So they decided to generate breast cancer organoids and perform a massive drug screening of multiple drugs that were available to treat cancer patients in general, and identify a combination of the mTOR inhibitor everolimus and the inhibitor of chromosomal segregation vinorelbine. And they treated the patient with this combination of drugs and identified that with this treatment, the disease was stable. So it's something that was not possible with the standard approach, that was with the standard chemotherapy approach, that was possible with this drug testing approach on breast cancer organoids. So really demonstrating the potential of this technology. And I guess I would like to finish my presentation by really discussing what are the current limitations for us to be able to develop these personalized medicine approaches in collaboration with potentially pharmaceutical companies. Well, I guess the use of this culture medium that we've been using for many years is patented. So we can use it for research purposes, but it's very restricted, and you can't really use it for commercial to establish commercial relationship with pharmaceutical companies. So how we can circumvent these limitations? And I guess this is why now the OncoPro culture medium can be very useful, because it doesn't really fall into these patents that were lodged in 2020. So what we wanted to do here in this context is compare the conventional medium versus the OncoPro medium and see whether they were able to perform similarly. So we used a triple negative breast cancer organoid line, the HBC14 line that we established using conventional medium. We plated these cells into the 3D matrix dome, into the matrigel. So we plated 30,000 cells per well. And we either use the conventional media, and I've listed growth factors below, or the OncoPro medium supplemented with beta-estradiol and FGF10. And we compare these two medium by looking at organoid morphology, number, and size. What you can clearly see, so this is the growth of the organoids over 21 days, comparing the conventional media and the OncoPro media. You can see that with both medium, we can successfully maintain the growth of the organoids over 21 days. If anything, the main difference between the two media, a small difference, is that with the conventional media, there was higher number of organoids that were generated compared to the OncoPro medium. However, the size of these organoids was similar between the two culture media. We've also performed a similar type of work, this time using colorectal cancer organoids, because within the Monash organoid program and the Monash Biomedicine Discovery Institute, we also have a very large biobank of colorectal cancer organoids that was developed by Professor Helen Abud in collaboration with Professor Paul McMurrick at Cardiff Hospital. And as you can see here, we use a colorectal cancer organoid line 73T that harbor mutation in APC and KRAS. We plated 20,000 cells per well. And as you can see in both conditions, we can sustain the growth of the organoids. And it's the phenotype is pretty much the same. We can sustain organoid growth, but there's similar size of organoids with slightly decreased organoid number with the OncoPro medium. Finally, when you look at potential morphological similarities between the conventional medium and the OncoPro medium, they appear to be very similar for breast cancer organoids and colorectal cancer organoids. And I agree with you, these are just pictures of organoids. And what we are aiming to do in the future is perform an in-depth characterization of these organoids. First of all, can we maintain the organoids for long-term and multiple passages using the OncoPro medium? Given the previous presentation by Colin, I would say very likely. Can we maintain similar cell populations between the two medium? It's something that we'll be looking at by performing, of course, RNA sequencing, but also histological characterization. But I believe that this medium is really, at this stage, it's looking very, very promising to sustain the growth of the organoids. So to conclude, I would like to thank all the people that are involved in this work within the Monash Biomedicine Discovery Institute, Dr. Eglantine Balin, the manager of the organoid program, and Dr. Stuart Archer, our bioinformatician, within my lab. And I would like to thank Dr. Jasvina Kaur and Dilis Lung that are very essential in driving this breast cancer organoids bioresource, and finally all our clinical collaborators at Cabernet within the Department of Surgery and within the Department of Medical Oncology. And to conclude, and to give a little bit of publicity for the Monash Biomedicine Discovery Institute and the organoid program, I would like to mention that we'll be organizing a symposium on emerging technologies in organoid research. This symposium is really method focused. So if you're interested in knowing how to grow organoids, how to measure cell viability, how to image, how to embed the organoids for histological characterization, but also what are the new tools to study, cellular interactions like co-culture or assembloid, this symposium is for you. This symposium will be organized on Monash University, the Clayton campus in Australia. It will be in person on the 6th of November, 2024. So thank you very much for your attention. I'm very happy to take questions.
Dr. Jarde, thank you so much for that informative presentation. It is now time for our live Q&A portion of our webinar. Now to our audience, if you haven't done so, please do so now. Submit those questions into the Ask a Question box. Click Submit, and we will answer as many of your questions as we have time for. So Dr. Jarde, if you don't mind, I'm going to dive in. We have so many questions already that have come in. So I'm going to start with this one. In your lab, do you see any significant differences establishing organoid lines from normal versus tumor samples?
That's a very good question. Well, what is counterintuitive in general is that the organoids established from normal tissues tend to grow more rapidly than organoids established from tumor samples. And we've seen that across multiple different tumor types, breast or colorectal cancer, and it seems to be a really very specific features. Probably also because when you look at the composition of the culture medium, it was really established initially for normal tissues with the normal biology in mind and not necessarily the cancer biology in mind. So it's very likely that we are using these culture conditions very promoting normal growth and unlikely, still promoting cancer growth, but not to the same extent. There's no massive difference in terms of normal organoids and cancer organoids in terms of success rates. And well, obviously the morphology and the mutation profile differ completely. But I guess in terms of feasibility of establishing these lines, as long as you apply the right growth factor medium, you should be able to establish them. The key point is also to use the right matrix for your work.
Thank you. Now our next question is two parts. Have you experienced normal healthy cells in the heterogeneous population outgrowing and overtaking the breast cancer organoids? And if so, what advice do you have for enriching cancer organoids?
Well, this one is a very tricky question. And we've been thinking a lot about this over the last 5 years, really. It's not really an issue, for example, if you work with colorectal cancer, because the main difference between establishing normal colon organoids and cancer organoids is the culture medium that you need to sustain the growth. In a normal colon tissue, to generate organoids, you need to use the WNT growth factor that is known to activate the WNT signaling pathway. However, in the cancer context, in colorectal cancer context, you don't need this WNT ligand because it's already activated. So you can basically remove this reagent from the mix and get rid of pretty much any contaminating normal cells that you would find in the primary colorectal tumor. Therefore, you really end up with a pure colorectal cancer organoid line. For the breast, it's very different, because currently the medium that we are using, that was established by Sachs et al., is suitable to grow normal breast cells and also breast cancer cells. And there's no way we can, and there's no difference between the medium. So we don't really play with a specific selective approach here. And we've been struggling with that to know exactly what we are growing. So I guess, if we go back to my presentation, what is essential, I think, is to perform whole exome sequencing in order to identify whether indeed you have a line with that contain mutation profile observed in the primary tumor. In order to select for, cancer cells versus wild type clones, we haven't found a good way to do that. But I guess what we want to do is also explore the oncopromedium, because according to the previous presentation, you can use that to sustain cancer growth, but not sustain normal cells. So it's something that we're gonna explore.
Excellent. Thank you for that answer. Our next question. Besides drug screening with breast cancer organoids, has your lab also tried conducting drug toxicity experiments on adjacent normal tissues to better study the survival rate and mutation frequency of these tissues, thus allowing further insights into drug efficacy, pharmacokinetics, and potential adverse drug reactions in a treatment plan?
Mm-hmm. Yeah, it's a good question. What we do every time we generate breast cancer organoids, it's to also establish normal breast organoids from the same patient in order to have a control for the breast cancer organoids. So the point here is to make sure that if we identified specific vulnerabilities of breast cancer cells, that this doesn't impact normal cells. So we use that as we use these normal organoids as a negative control. I guess if you're talking about systemic impact of potential drugs, I guess what would be ideal is to generate organoids from multiple of different organ types and assess the toxicity of these drugs on these organoids, because we know that when patients are undergoing chemotherapy, there's a potential impact on the liver, there's a potential impact on the kidney, there's impact on, for example, the skin or the small intestine as well. So you could potentially establish organoids from these multiple tissues and test these drugs on these normal organoids and see whether they do induce a phenotype or whether they are not toxic to these tissues. So having a more global approach, I think in the future would make sense as well, not necessarily focusing solely on breast cancer organoids.
Thank you for that. Our next question. What skills do a I'm having a rough night tonight. Required to work in an oncology lab, apart from scRNA-seq, how does scRNA sequencing help in cancer research?
Well, I guess, to be honest, I think it's essential now, given the development of all the technologies. So we're talking about whole exome sequencing, talking about single cell RNA sequencing, spatial transcriptomics now, that bioinformatics are embedded into teams. And within the teams, of course, you've got the bioinformaticians, you've got also the people working at the bench, and also the clinicians. So it's a very complete team that work together, and it's essential to translate this fundamental discovery into translational discoveries. So I guess single cell RNA sequencing can really help you in this context. If you're really interested in epithelial, the cellular composition of your primary tumor, and whether they are matched in the organoids, you could look at single cell RNA sequencing of the tumor and their matched organoids. So this would tell you whether, of course, you've got similar populations, and whether the organoids are a very good model to mimic the primary tumor. I guess on a general, if you've got a general approach, using single cell RNA sequencing is a fantastic technology that you can use to, understand interactions between all the different cell populations, and what are the crosstalk between these cells, and how you can leverage that to identify potentially new drugs. So bioinformaticians, and if you can develop skills in bioinformatics, this is definitely the future for research. There's massive needs in bioinformatics. And that's why we basically have multiple bioinformaticians working in my team and in the organoid program team as well. I don't know if it's good enough, but that's my answer. Yeah.
Thank you. So, Dr. Jarde, what are your thoughts to co-culture models of breast cancer organoids for screening assays? For example, co-culturing with cancer-associated fibroblasts or immune cells like NK and macrophages?
Well, there's I think it's very essential. So what I didn't really mention in my presentation today is that the organoid technology is very unique technology, but one of the limitations, it's important to know the limitations of methods, is that you grow solely the epithelial layer of the tumor. And if you want to basically develop models that are more complex, you need to add this cell population within or with the organoids. And indeed, if you want, you could potentially do co-cultures with cancer-associated fibroblasts with the organoids and see the impact of the cancer-associated fibroblasts as on the epithelial cells. That's perfectly fine, but you need to rebuild this cellular complexity. And I guess when you start thinking about these new models that are coming up, what I call the organoid 2.0 is really being able to generate assembloids, where these assembloids combine multiple cell populations. And we know that if you increase the number of populations that you mix within your organoids, they are more likely to resemble the primary tumor and the molecular features of the primary tumor. Consequently, they're also more likely to represent, the way the patient tumors are going to respond. So I think we need to move a step closer to developing complex models with multiple cell populations. We are currently working with cancer-associated fibroblasts and generating assembly model. You can also use immune cells, and I've seen multiple colleagues presenting movies showing NK cells or macrophages that were co-cultures with fibroblasts. So all these approaches are feasible, and we need to basically start combining many more populations to potentially in the future generate a complete tumor in culture.
Now, Dr. Jarde, we're almost out of time, so I'm going to wrap with this final question. You are seeing some early data with the OncoPro tumoroid culture medium. In general, can you comment on what's important to you when choosing a media system to establish patient-derived cancer organoids?
Well, to me, you want to make sure that first of all the medium that you're selecting is indeed supporting the growth of the organoids, and that's what we've observed so far on our primary data with the OncoPro medium. And indeed, you can sustain the growth of breast cancer organoids and colorectal cancer organoids. So it's really a first point. You also need to know what are the limitations associated with the medium, and the traditional medium that we use includes growth factors like aspartame, noggin, ureglin-1, EGF, and these growth factors and the composition of the medium has very strong patents associated with it. So it restricts the use for commercial relationship. So but it doesn't restrict the use for research purposes. So you also need to know the limitations that are associated with the culture medium that you're using. And this is not the case for the OncoPro medium. So you can potentially use it for research, and you can also use it for establishing commercial relationship with companies. I guess, what is also nice is simplicity of use, because generating, breast cancer organoid medium is quite challenging. You've got multiple growth factors, and if you have an organoid program that can support you within your institute, it makes things much easier. But if you don't, then it's quite challenging to make all these growth factors. So I guess having a ready-to-use solution like the OncoPro can really save time.
Very good. Dr. Jarde, thank you for this excellent Q&A session. Do you have any final comments for our audience?
Look, it was a real pleasure to be here with you today. I hope you enjoyed the presentations, and that if you have any questions or you want to follow up, please feel free to join to look at the Monash Biomedicine Organoid website. You can find our email addresses and send us an email if you want to start collaborating or discussing with us the organoid technology. That's it. Really have a good day.
Thank you. And I do want to thank Dr. Colin Paul also for his time today and his important research. We want to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, I do want to thank our audience for joining us as well and for their interesting questions we did not have time to get to today, and those submitted during the on-demand period. They will be addressed by our speakers via the email address you provided at the time of registration. And there were quite a few questions, so don't fret, we'll get those answered via email. Today's webcast also can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. To all our friends out there, thank you for joining us. Take care, be well, and have a great day. Bye-bye.
Thank you. Bye.